Assalamu alaikum. In this lecture, we will discuss the cell membrane transport. At the beginning, I will describe the cell membrane structure, and then we will discuss the ways by which the substances, whether molecules, electrolyte, substances, can cross the cell membrane. This is what is meant by transportation through the cell membrane. Let us talk about the structure of the cell membrane. The general characteristic feature of the cell membrane, it is thin and it is mechanically weak. As you can see, thickness is around 10 nanometer. So it is very thin and it is mechanically weak. It means it is easily be broken. This is the diagram of the cell membrane, and as you can see, it consists of two layers. This is the yellow layers made of lipid. That's why the cell membrane is described as a lipid bilayer membrane because it consists of two layers of lipid. Also, in the lipid, we have a protein. As you can see, these are protein, and this is also protein. And we have carbohydrates. The composition of the cell membrane mostly consists of protein, 55%, and then lipid, 42%. Carbohydrates, very little, as you can see, few carbohydrates attached to the cell membrane. As far as the cell membrane, the nature of the cell membrane is concerned, this cell membrane is permeable to certain substances and impermeable to other substances. It allows lipid-soluble material to cross the cell membrane, and the lipid-insoluble membrane cannot cross easily. It must be crossed through other means, not directly through the cell membrane, through protein channels or other means. We will describe these means later on. Let us talk about the other component of the cell membrane, the protein and the carbohydrate. Proteins are found into two forms. One is called integral, this one and this one, and they penetrating the cell membrane. And the other one is called peripheral. They are attached to the integral protein. They are not penetrating the cell membrane. Okay, mostly these protein attached to the carbohydrate. What about the actions of protein? Mostly the protein in the cell membrane act either forming a channel, this is a channel, or forming a carrier protein, this is a carrier protein, or enzymes here or there. And these enzymes are performing certain functions essential to the cell. The carbohydrate, 3% of the cell membrane is carbohydrates. We have two types of carbohydrate in the cell membrane. One is called glycoproteins. They are carbohydrate attached to the protein. And the other one is called glycolipid. They are attached to lipid. Both of them, they are called glycocalyx. The actions of the carbohydrates, very important. First of all, they act as receptors here, receptors in the cell membrane for substance to attach to it. And the other important function is sharing in the immune reaction. This is important for antigenic property of the cell. Each cell has certain character, and this character it is determined by the nature of the carbohydrates in the cell membrane. The other functions of the carbohydrate, they help in attachment of one cell to another. And the other one, or the other function, is providing negative charges to the cell. Why do we care about the cell membrane? Cell membrane is very, very important structure in the cell. And if this cell membrane breaks, the cell will die. And the other things about the cell membrane, it forms a partition, a barrier, hedges between the extracellular fluid and intracellular fluid. And as you can see, the composition of these fluids compositions are quite different. And what keeps this difference is the cell membrane. And as you can see, the concentration of ions and substances in extracellular fluid is quite different from intracellular fluid. That you don't need to remember the numbers, but you have to know how much sodium outside in comparison to the inside. In other words, بعبارة أخرى إنه we can say sodium outside is much more than inside, while potassium inside is much more than outside. You can see the calcium outside is much more than inside. Chloride outside is found in large quantity compared to the inside. Phosphate, mostly, it is found in large quantity inside, and so on. The pH inside the cell, the pH is around 7, outside around 7.4. The total amount of ions is called osmolarity. Total amount of ions here outside the cell, and here inside the cell are almost similar. يعني the quantity, how many ions here inside and outside are similar. We have many ways by which the substances can cross the cell membrane, and these ways are listed here. The first one is transportation by diffusion. We will talk about this diffusion later on. Next slide. We have two types of diffusion. One is called simple, the other one is called facilitated diffusion. The other way or other means for transportation is osmosis. This is for water. The third way is active transportation or active transport. And we have two types of active transportation. One is called primary active transportation. The other one is called secondary active transportation. And the last one is transportation in vesicles. And we have two types of transportation in vesicles. One is called endocytosis. The other one is called exocytosis. We will describe the transportation of material through the cell membrane by exocytosis and endocytosis first, because it is very simple. When we said exocytosis, it means transportation of material in vesicle from inside to outside, as you can see. Vesicles is formed here, as you can see, and these are the materials. And by binding the vesicles to the cell membrane, these substances can be thrown outside. يعني تخرج إلى الخارج. This is called exocytosis, and as you can see, through formation of vesicles. Endocytosis, the direction of crossing of substances, is the opposite of exocytosis, from outside to inside, but again through formation of vesicle. As you can see, material or the cell membrane is invaginated first and then forming a vesicles, and this vesicles will travel in the cytoplasm, and these vesicles, the cell or the membrane of the vesicle will be broken, and these material will be liberated to the cytoplasm. So we can say by endocytosis the materials are transported from outside to inside. Transportation through diffusion process. Diffusion as a term means random movement of particles in a solution. When we say diffusion, similar, the diffusion or movement of material through the cell membrane in random movement. When we have barrier as a cell membrane and particles at one side in large concentration, and on the other side the concentration is lower than the other side, the net movement of the substance will be from high concentration to low concentration, as you can see. And the movement of the particles will continue until the concentration of particles is equals on both sides, here and there. By the way, in this status of equal concentration, still there is diffusion of substances from here and there, but the net movement from here to here and from here to here is zero. In other words, the concentration will be the same on the same side of the cell membrane. Okay, and this type of transportation through the cell membrane does not need direct energy. We don't need to apply energy. The energy is coming from the kinetic energy of the substance itself. Here, the kinetic energy of the substances, because the concentration is high, is higher than the kinetic energy there. That's why the net movement, the movement of the material from one side where the concentration is high to low concentration will be more. We have two types of simple diffusion. First one is directly through the cell membrane, crossing the lipid bilayer cell membrane. And this type of diffusion is used by lipid-soluble substances like gases. Sorry, all the gases can transported through the cell membrane by this way, directly through the cell membrane, like oxygen, carbon dioxide, nitrogen, and so on. The other way is through protein channels, as you can see in this diagram. And this method is used by substances which are water soluble and lipid insoluble molecules like electrolytes and so on. This is a slide about diffusion of material directly through the cell membrane. And as I said in the previous slide, all lipid-soluble materials can pass directly through the cell membrane, like gases, oxygen, and carbon dioxide. The degree of diffusion depends on the lipid solubility. The more lipid solubility of the substance, the more the diffusion. So if you take carbon dioxide and oxygen, carbon dioxide lipid solubility is more than oxygen. For this reason, diffusion of CO2 is better than oxygen through the cell membrane. Other substances which are fat-soluble vitamins, which they are called lipophilic substances. The other name, hydrophobic substances like fatty acid, fat-soluble vitamins, and the drugs can dissolve directly in the lipid bilayer of the cell, and they can cross the cell membrane. Water can cross the cell membrane very easily, either directly or through certain channels. Diffusion of material or materials through protein channels. We will talk about sodium and potassium because they are found in large quantity in our cells. They are passing through simple, simple diffusion through the protein channels. These channels have two characteristic features. First of all, selectivity. It means they are selectivity allow one ion and they prevent the other ion. And the other characteristic feature, they are opened or closed by gates, as you can see, gates here and gates here. The gates can be opened or closed by many factors, and these are two important factors by which the gates can be opened or closed. One is called voltage gated channels. It means these channels can be affected by voltage, by electricity. The other one is called chemical or ligand gated or gating channels. These are affected by binding of a chemical to the protein or to the gate, either open it or close it. Once these channels are opened, the sodium or potassium will pass from high concentration to low concentration. In case of sodium, as you can see, from outside to inside, simply because sodium outside is more than inside, while potassium movement will be from inside to outside for the same, from high concentration to low concentration. Let us talk about voltage gated channels. An example is sodium gated channels. Sodium voltage gated channels. These channels are opened and closed by changes in electricity across the cell membrane. For sodium voltage gated channels, they are closed when the resting membrane potential is highly negative, as you can see, minus 90 millivolt. And they are activated and the gates are opened when negativity is decreased. Here is less than minus 90 to plus 35. The gates are opened. While potassium voltage gated channels are opened when inside become positive. For instance, become plus 35, it will be opened or they will be opened. The ligand gated channels, from the name ligand, it means chemical substance. And these chemical substances in our body, we have many types of them. One of them is called acetylcholine. For instance, in this case, if this is acetylcholine, this is a substance, chemical substance. And this chemical substance, once it reacts with its receptor or its place in the gates, it will activate the gates and it will cause opening, changes in the shape of the gates. In this case, it causes opening of the gates, and as you can see, once the gates is opened, the material will pass from high concentration to low concentration. If the ligand or the chemical substance is not attached to its receptor, the gate is closed. I put the acetylcholine channel because this is a common ligand or chemicals used in our body to open or close the channels. Facilitated diffusion. It means diffusion which needs help, and the help is through the carrier. And that's why this type is called carrier mediated diffusion. and this carrier is found in the cell membrane, as you can see in this diagram. You can see the molecule attached to the carrier, and the carrier changes its shape, allowing to release the substance to the other side of the cell membrane. So it needs a carrier protein. And these carriers, because they are protein, they have certain capacity. That's why it can be saturated. And if you see here this curve or the diagram, this is simple diffusion. The concentration of the substance and the rate of diffusion is directly proportional. But for facet diffusion, again, it is proportional to the concentration, but it will reach to its maximum, and with an increase in the concentration of the substance, there will be no further increase in the transportation, simply because it is saturated. The other characteristic feature of this type of diffusion or transportation, ATP energy is not required. Okay, so it is like simple diffusion, and they have specificity. Each substance has its own carriers. Best example for this type of transportation is the transportation of a glucose and amino acid into muscle and fat cell. For glucose, the insulin hormone increases facet diffusion simply because it increases the number of the carriers, and insulin can increase the glucose transportation by facilitated diffusion 10 to 20 times. This slide is about factors affecting the net rate of gaseous diffusion through the cell membrane. And these are the factors. And you can see in this equation, it explains everything about these factors. Diffusion is directly proportional. It is increased by increase in the total area of the membrane, increase by lipid solubility, and increase by the difference in the partial pressure between the two sides of the membrane. And diffusion is indirectly proportional to the thickness. It means the more the thickness, the less the diffusion, and to the molecular weight, square root of the molecular weight. The more the molecular weight of the substance, the less the diffusion of the substances. And we have another factor for affecting the net rate of diffusion of other substances, and for instance, electrolyte and so on. We have effect of concentration difference. The more the concentration difference, the more diffusion rate. Effect of electrical difference, this is a voltage, and also effect of pressure differences. Osmosis describes the movement of water through the cell membrane by diffusion, okay? And water is moving through the cell membrane very easily. And the factor affecting the water movement by diffusion or by osmosis depends on the concentration of solute on both sides. If you take here the solution or the solute concentration is low, and the concentration of the solute here is more, sodium chloride here more. These are big molecules of sodium chloride. So you can see water is moving from low solute concentration to a high solute concentration. If you take the situation from water concentration point of view, here the water concentration is more than here. So water is moved from high water concentration area to low water concentration area. These are a few points about osmosis of water. For water movement by osmosis, we need what is called semi-permeable membrane. In the semi-permeable membrane, allowing movement of certain material and does not allow other. In case of the cell membrane, it is permeable to water, but it is not permeable to solute, impermeable to solute. So this this condition is applied to the cell membrane. Also, osmosis is similar to diffusion, and if you remember the diffusion movement of material from high concentration to low concentration. So if we take this here, the concentration of water here is higher than there. So water will move in this direction, simply from high water concentration to low concentration. Water movement through the cell membrane is very important because at cellular level, the cell either shrink or swell by movement of water. And at tissue level, osmosis is needed for movement of water through the membrane of the gastrointestinal tract and renal system. And also, it occurs through the capillary wall. Active transport. When we say active, it means that we need energy. And usually, in this type of transportation, the ions or molecules are transported across or against their concentration gradient. In other words, في عبارة أخرى from low concentration to high concentration. The other characteristic feature, it is a carrier mediator or mediated. It needs carrier. We need carrier to carry the molecule. Uses energy. This energy either a direct ATP or directly from hydrolysis of ATP or indirectly from hydrolysis of ATP, as we will see just in a few minutes. Examples, we have many examples for this transportation: sodium, potassium, calcium, hydrogen, chloride, iodide, glucose, amino acid. All these substances can be transported against their concentration gradient. And this active transportation can be divided into two types, two major types. One is called primary active transportation. The other one is called secondary active transportation. The difference between these, the primary, the energy is derived directly from hydrolysis of ATP. While in secondary active transportation, we need energy, but this energy is use concentration gradient of certain substance, which is used, of course, by uses ATP as a source of energy. And best example for these, we have two types, by the way. One is called co-transportation. Secondary active transportation, usually it consists of two transportation of two molecules. So it is divided into two types. One is called co-transportation, mean that the solute are going in the same direction, both sodium and glucose moving from outside to inside. This is called co-transportation. If the movement of the two solute opposite, it is called counter-transportation. Sodium, calcium, counter-transportation. Sodium is moving from outside to inside, calcium from inside to outside, opposite to it. The primary active transportation, we have so many examples. The base example or the common type of this transportation in our body is sodium-potassium pump. Calcium, primary transportation, also very important. It moves the calcium from inside to outside, okay? The active transportation of hydrogen is found in gastric gland and in distal collecting tubules of the kidney. Sodium-potassium pump or transportation. It is the best example of active transportation. It is a carrier protein, or it needs a carrier protein, and it transports sodium and potassium against concentration gradient. It means from low concentration to high concentration. That's why we need energy in form of ATP. And as I said, it is a carrier mediated transportation. That's why it can be saturated. And it is very specific because it is specific to transport sodium and potassium pump. So let me explain this important transporter or transportation. This is the carrier or the protein responsible for movement of potassium and sodium. Here you can see 3 ions of sodium attached to the carrier, and these 3 ions of sodium will be pumped against concentration gradient from inside to outside. At the same time, 2 potassium ions are attached to this carrier, and these 2 potassium ions will be pumped to inside where a lot of potassium there. So potassium is pumped against concentration gradient from low potassium to a high potassium area. And this pump needs a lot of energy in form of hydrolysis of ATP. For example, cells lining the renal tubules use almost 90% of its energy for this pump. This pump or this transportation is very important because it is responsible for creating and maintaining high potassium inside and low potassium inside. And this makes the cell resting membrane potential. Also, this pump, by creating low potassium concentration inside the cell, this will be used as energy for secondary active transportation. We will discuss it in the next slide. Also, the last function of this pump is to prevent the cell swelling, because always it pulling or taking ions from inside to outside. Secondary active transport, we have 2 types: co-transportation and counter-transportation. Best example for counter-transportation is sodium glucose co-transport or sodium amino acid co-transportation. And because it is, or the movement of material in one direction is, sometimes it is called symport, okay? And we need a carrier for this, and the energy for this, or energy used by this transportation, is coming from the concentration gradient of sodium. So let us see this sodium glucose or amino acid co-transportation. This is the carrier, and you can see this carrier has 2 sites, one for sodium and one for glucose or amino acid. And by binding these substances to their sites, carrier changes its shape, and by this will bringing the sodium and the glucose from outside to inside. So they are moving in the same direction. This is a co-transportation, sim transport. The energy used is sodium concentration gradient, because here sodium is present in high concentration, here in low concentration. Glucose is moving against concentration gradient. That's why it is active transportation, because it moves glucose from low concentration to high concentration. The other type of this transportation is sodium hydrogen co-transportation, and this is found in the renal proximal tubule. Here you can see this is the urine, and sodium is moving in this direction and hydrogen is moving in this direction. Sodium is moving with the concentration gradient because sodium here is low, sodium here is high. Hydrogen is moving from high concentration to low concentration. This is hydrogen here high, hydrogen here outside is low. So this is active transportation. This slide summarizes everything about active transportation and different types of transportation. So let us see these important ones. This one, sodium potassium pump. We describe it in details. By the way, all these type of transportation needs carrier, need energy, and they can be saturated, and they transport material from low concentration to high concentration. So this is sodium potassium pump. This one is sugar and amino acid co-transportation. This one is sodium hydrogen counter transportation. This is potassium calcium counter transportation, and other transporters which are important in the cells. This slide shows the comparison of simple diffusion, facet diffusion, and active transportation through different properties. This columns showing the property of the transportation, whether it requires specific protein, membrane protein, whether it is highly selective, whether the transportation is saturated when the concentration is increased, whether the transportation is regulated by hormones, and whether the transportation is against concentration gradient of material or not, and lastly, whether the transportation requires ATP energy or not. If you come to this column for simple diffusion, you can see simple diffusion, always all these properties are negative to it. You can see simple diffusion, there is no requirement for protein. It is not highly selective. Sometimes it is selective, but it is not highly selective. It does not show transport saturation. It is not regulated by hormone, and it is not moving the material against concentration gradient. It moves the material with the concentration gradient from high concentration to low concentration. And lastly, it does not need energy. If you go to active transportation, you can see it meets all the properties here, positive properties. It requires protein, selective, it is saturated, it is hormonal regulated, it moves material against their concentration gradient, and it needs ATP activity or ATP energy. And here for facet diffusion, for these, the last two, facet diffusion, because it is diffusion, it moves the material from high concentration to low concentration. So there is no uphill transportation. There is no movement against concentration gradient. There is no need for direct ATP energy, but It needs protein carrier. It is highly selective and it can be saturated, and it is some of these transportation needs hormone. The best example is the insulin. If you remember, insulin is increasing the facilitated diffusion of a glucose inside the cell from high concentration to low concentration through carrier. So insulin is needed for facilitated diffusion of a glucose from outside to inside.